Welcome back, friends. In this video tutorial we'll be talking about Escherichia coli and especially the introduction about the Escherichia coli. Now Escherichia coli is the most common or commonly known microorganism for scientists because we have been studying Escherichia coli since the dawn of microbiology, and it is been going on. And Escherichia coli is a one of a kind of microorganism whom we know most of the properties, right? Now, usually Escherichia coli, if I want to talk about Escherichia coli, it may take a longer time, but in the short span of the video, I'll be talking about some special features about the Escherichia coli and among the Escherichia species also, because Escherichia species is a kind of uh, microorganisms that are living in our gut, and these microorganisms are simply short rods. So let me write about their properties. They are short rods. So begin with single properties. They are short rods, okay, and also they are facultative anaerobes. They are facultative anaerobe. So let me facultative anaerobe, because they live in our gut, they won't require oxygen for their growth in this case much, okay. And the most important property above all, they can ferment lactose. So they can ferment lactose, and this particular feature makes them totally distinct from rest of the enterobacter species which are living inside our gut, because the gut living bacteria, among the gut living bacteria, we are having Escherichia coli, we are having Salmonella, and we are having different enterobacter species, but among all of them nobody can ferment lactose except Escherichia species, okay. Among the Escherichia species, Escherichia coli especially, they can ferment lactose, okay. And usually all of them can ferment glucose, but this one can ferment lactose, okay, because they are having the lactose fermenting ability containing gene. It is called the lac operon concept. So lac plus will be termed for the Escherichia coli, okay. And usually they are catalase positive, catalase taste positive. What is catalase taste? If you want to find, you can go in my YouTube channel, you will find it in the previous videos of this bacteriology session. So catalase positive, but they are oxidase negative, oxidase negative, okay. And definitely they are gram negative. I should have mentioned it earlier. Escherichia coli is a gram negative. They are gram negative type of organism. They are also motile with their flagella, and they are having the capsule layer, which is a slimy layer coating this whole bacteria. So capsule layer is also there, okay. And we can culture them in McConkey agar. It's a special kind of medium, McConkey agar. So you can culture them on McConkey agar pretty easily, okay. So these are the basic quick properties about Escherichia coli. But the most important thing is that Escherichia coli live in our gut, and this is very in normal times. When they live in our gut, they act as a beneficial microorganisms in most aspects, and they are also known as a friend of microbiologists, because we use Escherichia coli in laboratory for studying different bacteriological effects or cellular events of bacteria using Escherichia coli as a model organism, okay. And in our intestine they can act as what you can say, so positive mediators, or they are leading to the positive effects, but sometimes they can cause diseases. Some strains of Escherichia coli are pathogenic, but rest of the strains are non-pathogenic in nature. But those Escherichia coli that are present in our gut, sometimes they are not pathogenic, but when they are encountering and they are entering into our bloodstream, they become pathogenic. So they usually become pathogenic. So let me know. They usually become pathogenic. Usually become pathogenic when entering bloodstream, okay. So that is an important point. When they enter bloodstream, only they become the pathogenic in most of the cases. Otherwise they are non-pathogenic in nature. So this Escherichia coli can be divided depending upon their different antigen that are present in their outer membrane. And as they are gram negative bacteria, I should have told this earlier, that as they are gram negative bacteria, they are having outer cell membrane, outer cell membrane, right. Now this outer cell membrane contains lipopolysaccharide, right, or LPS. So what they are having, the LPS layer or lipopolysaccharide. So let me write lipopolysaccharide layer. Now this lipopolysaccharide layer is antigenic because this can serve as an endotoxin. This can serve as an endotoxin, right? And this lipopolysaccharide is made up with several different component. One of the component is O antigen, right? Now depending upon these different kinds of antigen that are present in lipopolysaccharide layer, we can divide Escherichia coli into different segments. And not only the antigen that is present in lipopolysaccharide, but also there are antigens that can be present in the flagella, antigens that can be present in the capsule, right? So depending upon those antigens, we can divide Escherichia coli. For example, in antigens present in LPS layer, we can distinguish them. They are called O antigen. O antigen. Now the antigen that are present in, let's say capsule, in a capsule layer, those are called K antigen. And obviously the antigens that are present in, let's say, present in LPS is done, capsule is done. Let's say the antigen present in flagella. In those cases, we call those antigens that are present in flagella as antigen H or H antigen. So depending upon the presence of this antigen, we can divide it into O antigen, K antigen, and H antigen. So depending upon this kind of antigens, we can vary the different name of Escherichia coli, right? So the nomenclature of Escherichia coli come from this kind of antigens. Now you may probably heard this name, that this strain O157H7, right? You probably heard this name because this is a potent pathogen for human being. Now this type of Escherichia coli strain is acting as, or they are called as, enterohemorrhagic E. coli, EHEC, or enterohemorrhagic E. coli. So they are very, very dangerous. They can produce toxins, and the toxin is made up with this kind of antigens. Now for this particular strain, as we can see, there are two different types of antigen. One is the O antigen. This is present in the lipopolysaccharide layer of this particular bacteria, and another one is the H antigen, which is the H number 7. So this antigen is present in the flagella. So depending upon the presence of this antigen, we term this O157H7. So this is a potential pathogen for human being. We'll be studying it later. Okay, so depending upon these antigens, we are having different names like that. Okay, so in a sense, that's the introductory part about the Escherichia coli, and I hope that's helpful. Thank you. Okay guys, so far we have talked about the introductory video about the Escherichia coli. Now in this particular tutorial, we'll be talking about the infectivity of Escherichia coli. So we'll be talking about infectivity of E. coli. Now, among the infectivity of E. coli, what we know is that there are several different types of E. coli depending upon the type of infection they cause. Now, usually there are four major types of E. coli depending upon the infection. Now, generally, all of these enterobacter type of species, or all these gut-living bacteria, they are related to any kind of gastrointestinal diseases, abdominal cramps, diarrhea, and all these different types of symptoms. More or less, the symptoms are same for all the different types of diseases, and so on. In case of Escherichia coli also, we can see those type of symptoms, right? But in case of depending upon the type of or the causative agent of the disease, so some of the Escherichia coli bacteria can cause the disease using heat labile toxin, some of them causing this disease using heat sensitive toxin, some of them causing this disease simply by hemorrhagic nature and all these things, and obviously by any kind of other means of this disease. Okay, so depending upon this disease, we can divide the Escherichia coli in four different groups. So let me write, so depending upon the disease, so let me write as E. coli. It is better known as E. coli. So E. coli, we can divide this E. coli into four different sections. And what are those sections? The first one I'm going to write as ETEC, or enterotoxigenic. So let me write enterotoxigenic E. coli. So enterotoxigenic E. coli, and it is termed as enterotoxigenic. So we'll be taking here, so let us take E here, T here, E here, and C here. So ETEC, It is termed as ETEC, or enterotoxigenic E. coli. Now the second type of E. coli that we know is enteropathogenic E. coli. So enteropathogenic. So let me write enteropathogenic E. coli. Okay. Now this one, so let us write the terms. Here it is E, here it is P, then we are having E and C. So EPEC, it will be termed as EPENC. So enteropathogenic E. coli. Now the third type of example is enterohemorrhagic E. coli. So let me write enterohemorrhagic. So the hemorrhagic, HEMO, hemorrhagic, RRHAGIC, enterohemorrhagic E. coli. So this is the third kind, and it is also called as EHEC. So enterohemorrhagic E. coli, or EHEC, the third kind. And the last kind, so let me write the last kind here, fourth kind I'm going to talk about, is enteroinvasive E. coli. Okay, and this one, so let me write it EIEC. So enteroinvasive E. coli, or EIEC. So these are the four different types of E. coli that we are going to talk about. Okay, so depending upon the type of infection they cause. Now the first one was or enterotoxigenic E. coli. It causes what we know as a traveler's diarrhea. Traveler's diarrhea. Sorry for the bad handwriting though. Traveler's diarrhea. Now for this traveler's diarrhea, what it does actually, it causes a kind of watery diarrhea, and obviously abdominal cramp is associated with it. In case of this enterotoxigenic E. coli, most of the time this kind of enterotoxigenic strain of E. coli, what they do actually, they are changing, they are elevating the concentration of cyclic AMP inside the cell. So as a result of this elevation concentration of cyclic AMP inside the cell of our endothelium lining inside our gut, those cells start to lose water, as well as start to lose other type of ions like chlorine ions outside. So as they're releasing water and chlorine outside, the gut becomes extremely fluid in a limited amount of time, so that it results in the very watery kind of diarrhea. Okay, and the reason for that is the continuous signaling of cyclic AMP, so the cyclic AMP, to on and on and on throughout the time, because you know that cyclic AMP is acting as a messenger during the signal transduction pathway, right? Now the signal that brings to cyclic AMP to rise the concentration of cyclic AMP will be conveyed by a kind of a signal receptor. Now in this case of this enterotoxigenic E. coli, they produce a toxin, right? So it's a toxigenic, right? So it produces a kind of toxin. So that heat labile toxin, so we call it an LT, or heat labile toxin. Now this heat labile toxin will go and bind with that receptor for that signaling, and it binds with it throughout the time, so that the signal is on, and it is telling this cyclic AMP concentration to go on and on and on and high, and as a result of that, it results in the release of water and chlorine ions outside into the gut, and the cell becomes dehydrated and the gut becomes extremely fluid, right? So that is the actual case of enterotoxigenic E. coli. And again, this kind of disease is mostly seen in all this kind of disease are related to hygiene, and they are mostly seen in developing countries where they are poor sanitation is there and all these things. Okay, so these things matter actually. Okay, now the second type of strain is called the enteropathogenic E. coli. Now this enteropathogenic E. coli is again causing watery diarrhea, and this watery diarrhea caused by enteropathogenic E. coli is for a long duration. So it's for a long duration, and also this kind of disease or this kind of condition is caused in a newborn. They are caused in newborn, mostly in infants. Why? Because in those cases, this kind of infection can be spread from their mother to the newborn, and obviously this is a result of oral fecal transformation. In both these cases, enterotoxigenic E. coli and also enteropathogenic E. coli, in both these cases, they causes this kind of colitis or this kind of diarrhea kind of symptoms. In all these cases, The reason for that is oral fecal. So let me write, it's oral fecal or fecal oral, whatever, fecal oral transfer of this bacteria, right? So the poisoning of this bacteria usually transferred via the water or food sources, right? So for both these cases, this is the reason. But enteropathogenic E. coli is persistent for a longer period of time, and it is affecting the newborn or the infants. Okay, and mostly they are seen in developing countries like India is an example for that. Africa is another example like that. Okay, now here it is, the third kind, the enterohemorrhagic E. coli, and this kind of E. coli is pretty dangerous. Remember, I've talked about a member of this enterohemorrhagic E. coli, and that member is uh, AOH, what is it, O157H7, right? So that particular strain is one of the EHEC member, and for this reason, this kind of Escherichia coli generally cause diseases using endotoxin, a kind of endotoxin. So endotoxin. Now this endotoxin we know is from the LPS, right? And this kind of condition, what it results in, the rupturation of blood, and it also lead to the what you can say, cut in our blood vessel. As a result, blood will flow along with the along with urine, as well as along with stool, and as a result it will result in the bloody diarrhea. Okay, that's why they are called hemorrhagic colitis, because in this case they start to break down the RBC. So RBC, so let me write RBC breakdown, RBC breakdown, and also blood vessel breakdown. As a result of that, red blood cells coming out, and it results in the bloody diarrhea. And this can also related with the urinogenital kind of region, and it is then called, the combinatory effect is called the hemolytic uremic syndrome, or HUS. It is called hemolytic uremic syndrome, or HUS, hemolytic uremic syndrome, or HUS. Okay, so these all things are coming from the enterohemorrhagic E. coli. So this is one of the most dangerous kind of condition. And the fourth one is the invasive E. coli. Now invasive E. coli, again it causes bloody diarrhea. So it again causes bloody diarrhea. So let me write bloody diarrhea. Okay. And the reason for that, again, they are producing a kind of toxin. Again, the reason is kind of toxin. And this kind of invasive E. coli and all these things, they can only be activated when they are transferred to the bloodstream. So normally when they are present in gut, they are non-pathogenic, but when they are transferred to the bloodstream, in those cases they become very much pathogenic and cause severe diarrhea. Okay, so in all these cases you have seen all this infectivity leads to most of the cases bloody diarrhea, and almost all the disease, the symptoms are called colitis, and they are related to diarrhea a little bit, not a little bit, but longer extent. And obviously sometimes they're related to hemolytic uremic syndrome, or that means blood is coming out with urea or with urine, so it's HUS is associated. So all these things are the part of it. Okay, so that's the infectivity about all the Escherichia coli, uh, most pathogenic Escherichia coli types. And obviously there are other kinds, but I'm not going to talk about them. So that's it, and I hope that's helpful. Thank you.
Welcome back, friends. We have so far talked about the introductory video about Escherichia coli, and also we have talked about the infectivity of Escherichia coli. And we have seen there are several different types of Escherichia coli infections. Depending upon the mode of infection they cause, we also term them differently, like enterohemorrhagic E. coli, enterotoxigenic E. coli, enteropathogenic E. coli, and all these things. Okay. Now, among all of these different types, so let me write those types. So we'll be talking about here in this video. We'll be talking about pathogenesis, or the property or the mode of infection that is taken by Escherichia coli. And among all this pathogenic version of E. coli, so let me write the pathogenic versions of E. coli, and those versions are enterotoxigenic E. coli, or ETEC. Then we are having enteropathogenic E. coli, or EPEC. Then we are having enterohemorrhagic E. coli, and also we have talked about the invasive E. coli. So these four different kinds, and we have seen they're associated with different kinds of diseases, but almost all of this group are causing diarrhea, right? So we have seen that almost all of them causes diarrhea. So diarrhea means simply loss of water, loss of water from intestinal cell, from intestinal cells. So it's a kind of intestinal cells. And it's a kind of dehydration, right? We know that this is a kind of dehydration that we can talk about diarrhea, right? And always some of them causes simple watery diarrhea. So a lot of water is coming out with stool. Some of them are also causing bloody diarrhea. So let me write bloody diarrhea. I know in those cases blood comes out with stool and a lot of water, right? So in both these cases, some of and in all these cases, this part of the mm, reason or disease like diarrhea and this kind of symptoms are caused due to the presence of endotoxin, presence of endotoxin. Now the endotoxin, remember, the endotoxin that is present onto the surface of E. coli, and that endotoxin is called lipopolysaccharide layer. You all know that for gram-negative rods, we can find this lipopolysaccharide layer outside. Now, among these lipopolysaccharides, we are having antigenic determinants, right? We are having antigens. Now, the antigens we are having usually are called O antigen. We have talked about them. And also, this kind of bacteria can have H antigen in their flagella. And also, they can have other types of antigens, right? Now this, depending upon this O antigen, this can infect our host cell, right? And how they are infecting this, we'll be studying it in this video. Now this endotoxin that we're talking about, this kind of toxin can be of two different types. So let me talk about this kind of toxin can be of two different types. So one type is heat labile toxin. Another type is heat resistant heat resistant or let me write heat resistant or heat resistant or or heat stable kind of toxin. Now the kind of toxin heat labile, they are called as LT toxin, and those one which are heat stable are called ST toxin. Okay, so ST kind. So this LT and ST are depending upon whether they are heat resistant or heat labile. Okay, now all this kind of toxin, especially in this video we are going to talk about this LT type of toxin, because we're going to see the mode of activity of this LT toxin. Now you've seen that diarrhea is a massive loss of water from the intestinal cells, right? So let me draw the intestinal lining here for you. So let me draw the intestinal lining. So let's say here it is in the cross section. So we are looking at the cross section of intestinal cells. So let's say these are the villi, right? So villi is coming out like that. So this is the intestinal lining, right? And these are the intestinal cells are arranged, and these are the villi that are coming out, and here inside this is the gut, which is actually making a stool and all these things which will come out from this region, but it can have trouble. Now what is the problem? Let us look at here. In this case what we know is that these intestinal cells, they are having different transporters, remember, because if you're talking about cell membrane, so let me talk about cell membrane a little bit. If you're talking about cell membrane, that this membrane is permeable to few gases. So few gas can normally diffuse through the membrane, but except for those some gases like nitrogen, oxygen, and all these things, other molecules like say large molecules and ions must be passed through a channel, and there are channels present inside this in this cell membrane, right? Like embedded, they are called transmembrane channels like that. Now through this channel we are having different ions, like say sodium ion can pass through, potassium ion can pass through, chlorine ion can pass through, in or out, and all these cases like that. Okay, and this movement of ions, is depending upon a kind of chemical signaling. It is depending upon a chemical signaling. Now the signaling we're talking about here is involving, ATP and cyclic AMP concentration. It is depending upon the cyclic AMP concentration, the concentration of cyclic AMP in this case. Now this case, let me write. This is about the concentration of cyclic AMP inside the cell. Now, if the concentration of cyclic AMP elevates inside the cell, it will cause the release of sodium ion as well as chlorine ion massively from the intestinal cells into the gut. And if there is a what you can say, this cyclic AMP concentration is keep going down, in those conditions sodium and other kind of ions will pump inside this intestinal cell from the gut region, right? So simply this is the condition. So let us talk about it. Now in this case of this villi, there are cell membranes, and the cell membrane of these villi cells, they are consisting of several kind of receptor molecules. So let me draw one of the receptor molecule. Let's say this is a receptor. Let's say these are the receptor molecules I'm drawing right now. So there are receptor molecules that are present. Now these receptor molecules, let me write, they are receptor molecule and receptor for signaling, right? Receptor molecule for signaling. What kind of signaling we are talking about? We are talking about the signaling via via the signaling which is involving the G protein coupled receptor, right? The G protein coupled receptor. Okay. Now in this case, the signaling molecules are present, and whenever it is getting any kind of signal from outside, and that signal is telling these villi cells to release sodium ion, they will release, and they will release it if we are having a high concentration of cyclic AMP in this region. And if we are having low concentration of cyclic AMP in this region, in those cases they will release sodium and potassium and chlorine ion outside. So this is the basic process. Now in this case, what we can see, this heat labile toxin or LT kind of toxin, so let me write this LT kind of toxin, they move inside. So as a result of this LT toxin released by gut bacteria, so let's say these red things are the LT toxins. Now some of the LT toxin will go and attach to this receptor. So they bind with this receptor. As a result of this receptor and LT toxin binding, they will make they will trigger some secondary response inside the cell, right? And the response they trigger inside the cell is always on for a longer period of time. So usually whenever a kind of natural signaling molecule come and stuck to this receptor molecules, it will provide some signal, and the signaling will occur, cyclic AMP concentration rises, and sodium and potassium pumped sodium chlorine pumped outside into the gut, and then the signal stops because this molecule will cleave out and go away. But in this case, this LT or heat labile toxin won't be cut and go away. Instead, it will be still binding with this receptor, and it is binding throughout the time. As a result, it is keeping the signaling pathway on throughout the time. As a result of that, what we can see that as a result in this internal region of this villi cells, what we can see rising in the ATP concentration. So as we have raised the ATP concentration, this ATP will be not ATP, rise in adenylate cyclase, which is an enzyme. So let me talk about it. So rise in an enzyme, and that is called adenylate cyclase. Adenylate cyclase. Now this, as a result of the elevated concentration of adenylate cyclase, which is an enzyme to convert ATP into cyclic AMP. So the role of this adenylate cyclase is to convert ATP into cyclic AMP, and this is brought about by, remember, adenylate cyclase. Yes. Okay. Now that's the case. That's the usual case in this case. Okay. Now what we can see, as a result of this binding of LT toxin to the receptor, it will give the concentration of adenylate cyclase rising. And as a result of this concentration of adenylate cyclase is going higher and higher, it will convert more ATP into more cyclic AMP. So eventually we will be ending up with more and more cyclic AMP here inside. So more cyclic AMP inside, more cyclic AMP inside. So as a result of the concentration of cyclic AMP is getting higher, remember we have already talked about it. If the concentration of cyclic AMP is going higher, in those cases it will trigger the release of sodium, chlorine, and as well as water from this intestinal cell outside the gut. So now what we can see, in this case they will start releasing the sodium as well as chlorine as well as water outside. So sodium. So let me change the color here. So we are having sodium. So they kick this sodium, chlorine as well as water out from this microvilli cells inside the gut, and they will keep them flowing outside. So as a result, what we can see, we know that there are different kind of transporter present in cell membrane. Now as a result of this triggering mechanism, they keep on releasing water outside into the gut. So as a result, the gut fluid is become very watery, and obviously they are mixed with stool. So now we can look for the watery stool kind of disease, and it is eventually turned into diarrhea. Okay, and the cells, the microvilli cells, are now, as a result of lacking of water, they are becoming dehydrated. So we also talked about the dehydration in this case. So this thing is caused due to the prolonged activation of this signaling pathway via adenylate cyclase after the binding of heat labile toxin to the signal receptor. So this is a kind of technique that are taken by almost most of these E. coli cells. Okay, some of them also having slight variations among all this process, but more or less they are having a kind of similar things like that. Okay, so that's the pathogenesis, and I hope that's helpful. Thank you. Okay guys, so we have talked about the infectivity of Escherichia coli, and also we have talked about the pathogenetic properties of Escherichia coli. Now in this video we'll be talking about the treatment in a little bit amount. Treatment we'll be talking about. Now, in treatment part, usually this kind of infections caused by Escherichia coli, most of the case the infections are of systemic diseases like colitis or systemic diarrhea and all these things. Now, in all these cases, we know that Escherichia coli is obtained via food source, contaminated food source as well as water source. So we need to be very careful, very careful, very careful about choosing, very careful about choosing food and water source. Food and water source, that's very important. That's very important. And obviously a kind of disease like which is caused by enteropathogenic E. coli, mostly they are causing this disease to infants, right? So in those cases we need to be very careful about our hygiene and sanitary system and all these things. So in this case of whole Escherichia species causing infections, we need to be much more hygienic, and especially in the developing countries we need to be very careful about all these facts for choosing our food and water sources. And obviously the food are properly prepared, food totally boiled and all these things. We need to be very careful about all these things. So prevention is always better than cure, and especially in this case it is the most important thing. Now even if the infection is spreading, even if the infection is spreading any kind of uh, problem, in those cases we need to use antibiotics, because Escherichia coli is a bacteria, so we can use antibiotics for our treatment. Now the antibiotics that we can use for this kind of treatment, usually for the systemic infections, among the penicillin zone, among the penicillin type, we can use ampicillin, we can use ampicillin. Okay, and among the cephalosporins also we can use, among the cephalosporin, among the cephalosporins we can use, in those cases we can use cefotaxime. This is a kind of third generation cephalosporin, so we can use cefotaxime in this case also. And in case of UTI or urinary tract infections, which is also can be caused by Escherichia coli, some species of Escherichia bacteria. In those cases, we can use cefotaxime as a remedy in those cases. Okay, so this can be used in all these cases. Except for all these things, if we're talking about other kind of antibiotics, we can also use trimethoprim, trimethoprim or trimethoprim, whatever, or also uh, sulfamethoxazole, or we can use methoxazole. Okay, so you can use these things also. But most commonly we are using ampicillin and cefotaxime for curing this kind of infections. Because once this infections is spreading, E. coli infection is spreading, that means you are having watery diarrhea and running stool and all these things. In those cases, you need to be very careful about choosing food and water again. And also, you need to balance, you need to balance, you need to keep balance of electrolytes and water inside your body. Otherwise it will make trouble, because you are literally dehydrating. So you need to take a lot of water which is pure, which is definitely boiled if it is possible. And you need to be very careful about choosing the electrolytes, because in those conditions you can have ORS, or oral rehydration solution, to take. And in this case, this ORS is filled with all the electrolytes and sodium salt, potassium salts, and all these things that will help you to restore the electrolyte loss of your intestinal cells. And also you need to take a lot of water, right? And obviously you need to use this kind of antibiotics, because one kind of treatment will not help. You need to balance your electrolytes and take up water, as well as you're treating with antibiotics, and keeping all these things at the same time can limit the infection a little bit extent. Now, and all these kind of infections are can be treated pretty easily under 2 to 3 days maximum. For infants, however, it is very very dangerous. For infants or for newborn, I'm telling you, for newborn, let me change the color. For newborn, it is very very dangerous. So you need to be very careful about newborn, because in India many newborn die with the help of this kind of Escherichia coli infection. So you need to be very very careful for the newborns. So taking them right measures, their food must be cooked properly, their water source and everything. You need to be very very choosy for all those things. Okay, so that's all about the treatment of E. coli infections, and I hope that's helpful. Thank you.